Photosynthesis is when plants absorb sunlight, water, and carbon dioxide and convert it into glucose and oxygen. This occurs through two reactions, the light and dark reaction.
The light reaction takes place in the thylakoid.
First, sunlight is absorbed so it can split H2O molecules into H plus and O2 and produce excited electrons into the photosystem II, which leads to the electron transport chain.
The oxygen is released from plant through cellular respiration. The hydrogen will start to accumulate in the inner thylakoid.
The H plus that have accumulated the inner thylakoid enter an enzyme called the ATP synthase, which converts ADP into ATP. The electrons continue traveling through the ETC until they reach photosystem 1 and help convert NADP plus into NADPH.
The energy from electrons traveling through the ETC is used to pump H plus against the concentration gradient and into the inner thylakoid.
The ATP and NADPH produced during the light reaction are then transferred and used in the dark reaction. The cycle starts with an organic substance called RUBP, which has a 5-carbon atom.
The CO2 that was absorbed from the atmosphere is added to the RUBP to create a molecule with 6 carbon atoms. This is the carbon fixation stage.
The next stage is called reduction. NADPH comes in and causes the C6 molecule to break into two C3 molecules and then converts back into NADP+.
One of the C3 molecules will be converted into G3P, which will be made into glucose with the help of ATP. The ATP will be converted back into ADP.
The other C3 molecule will go through the regeneration stage. Another C3 molecule will be added to the original one, and with the help of ATP, it becomes an RUBP molecule again.
The ATP converts back into ADP, and the ADP and NADP plus released from the dark reaction are recycled back into the light reaction.
In this lab, we observe leaf chads that have been deprived of oxygen go through photosynthesis and float to the top as they release their own oxygen.
The tools you will need for this lab are baking soda, soap, syringe, some leaves, a hole puncher to cut the leaf chads out, cups, a timer, and a light source.
And to observe this, we first had to get our leaf chads to sink. To do this, we had to use a syringe and take a water solution with the chads and cover the top with our thumb as we pushed or pulled so the air inside would be released.
Keep doing this until all of the leaf chads have synced, and then pour it back into the beaker. Start your timer and record every minute how many leaf chads are floating. Do this until all the leaf chads are floating. In our general experiment, we compared photosynthesis of leaf chads with 2 grams of baking soda against 0 grams of baking soda. This is the beaker with only 2 grams of baking soda. It took a while, but we had all our leaf chads floating by 19 minutes. This beaker was the control group, meaning there was no baking soda in it. We recorded for an entire 12 minutes until we realized that none of the leaf chads would float because there was no carbon dioxide to help them perform photosynthesis. Observing these results, we decided to try out 3 more different concentrations of CO2. Our hypothesis was that the higher amount of CO2 in the water, the faster the leaf chads would produce oxygen and float to the surface again.
On the left is the beaker with 3 grams of carbon dioxide, which took about 10 or 28 minutes for all 10 chads to rise. On the right is the beaker with 1 gram of carbon dioxide, which took about 17 minutes for all 10 chads to rise. This is the beaker with 3 grams of carbon dioxide, which took longer than the beaker with 1 gram of carbon dioxide. We think it's due to the different light, which could have caused different temperature.
This beaker had 4 grams of baking soda and took a very quick 7 minutes to have all the 10 leaf chads floating. This was the control group, and as you can see, nothing has happened. This was the data for 1 gram of baking soda. It had a pretty constant rate of photosynthesis. For the 2 grams, nothing had happened for the first 11 minutes, but then it quickly spiked up for the rest of the 5 minutes. This was the 3 grams, which was sort of an abnormality to the rest of our data, since it took so long, and the 4 grams, which took the quickest out of all, with only 7 minutes.
Overall, the results of our experiment were as expected, with exceptions to the 3 grams, which we don't know why it was so slow.